All right, guys, welcome back to Underground Science. In this video, we'll be talking about the complement system again, but this time we'll be talking about the alternative pathway. Okay, so let's see how this works. So if we remember the classical pathway, you needed antibodies, right? Like IgG and IgM. But in the alternative pathway, this is antibody independent. Okay, this is antibody independent. Okay, what this means is you don't need antibodies for the alternative pathway to become activated and for it to work. Before we start talking about the alternative pathway, it's important to know that specifically C3b, okay, we'll see this later, but C3b, which we also talked about in the classical pathway, and I forgot to mention this, is that C3b can actually directly bind to the pathogen surface, okay, to the pathogen surface. So if we take any pathogen, for example, here, C3b can actually bind onto the pathogen surface directly, which can lead to neutrophils or macrophages, right, phagocytosing and destroying this pathogen. So that makes C3b a strong opsonid, okay, because it makes the pathogen more attractive to certain white blood cells like neutrophils and macrophages. Okay. And of course, we know that if macrophages are the one to take in and destroy the pathogen, it can also put certain parts of the pathogen onto MHC2, triggering the adaptive immune system, right? So I just wanted to talk about that before we started. So before we started the alternative pathway. All right. So how does the alternative pathway work if it doesn't require antibodies? Okay, so we have something called the tick over. Okay, and we'll talk about what this is. The tick over. So what the tick over is, it's its constant hydrolysis of C3. Okay, so if we take our bloodstream, which we know contains plasma, right? We have C3, right, floating. Okay, so let's say this is C3. Okay, so we have a bunch of C3 here. So if we just say that, okay, this is our C3 protein or C3 complement. We have C3 floating, and in our plasma, it's undergoing constant spontaneous hydrolysis, okay, at a low rate, of course. So what this means is we have this C3 that's constantly getting converted into C3 that's attached to water, okay? It's C3 hydrolysis. So we get something called hydrolyzed C3. So if we draw that, we have C3 here, and then to represent it, I'm going to draw this representing water. And the way we're going to denote this is C3H2O, okay? So this is hydrolyzed C3, okay? This is going to be our hydrolyzed C3. So this is constantly happening at a low rate in our plasma. This is called the tick over, okay? Now, once this happens, once we get hydrolyzed C3, the whole goal of this is so that we can start producing C3b. Okay, so once we have hydrolyzed C3, factor B will come next. Okay, so then we're going to have factor B in the play here, and we'll draw factor B in purple. So then we'll have something called factor B, and that's going to come and it's going to bind onto the hydrolyzed C3. All right, but once that happens, right, so if we say this is factor B, let's go and label this as factor B, then we said that that's going to bind onto our hydrolyzed C3. So let's go ahead and represent that. So we're going to have C3 here and our water here. And then we're going to have factor B right here, right? Once that happens, we're going to have another molecule called factor D that's going to come in. And this is actually going to cleave factor B. And it's going to cleave it into BA and BB, okay? So if this is our factor D, right, we're going to have factor D that's going to come in, it's going to cleave factor B into two different molecules. It's going to cleave it into BB, right, which will represent like this, and it's going to cleave it into BA. So this is going to be BB, and this is going to be BA. Now we don't care about BA, okay? So we don't care about BA. What we should focus on is BB, okay? Because that's gonna be the protein that's going to be attached to the hydrolyzed C3. So now we're going to have this. We're going to have our C3 that's hydrolyzed. So let's go and draw the water. And then we're going to have BB attached, which we drew it as a square, right? So what do we have here? We have, let's go and write what we have overall, right? Overall, we have C3 that's hydrolyzed with our BB attached to it. Now, this is called our fluid phase C3 convertase, okay? C3 convertase, this is our C3 convertase, but it's in the fluid phase because it's hydrolyzed, right? So it's in the fluid phase because it's hydrolyzed. Now, the reason why this is important is because this is the first active enzyme in the alternative pathway, right? What's going to happen is this is still C3 convertase. So C3 that's flowing in our blood, right, can actually come in and get acted by the C3 convertase. So let's go and draw that. So if we have C3 that's flowing around, right, and it comes in contact with this fluid phase C3 convertase, we're going to get C3A and C3B production. Now, C3b now can actually go and bind onto the pathogen surface. So if we have a pathogen nearby, right, like this, right, if we have a pathogen nearby and the fluid phase C3 convertase just converted C3 or just converted C3 into C3a and B, the C3b can go and bind onto the pathogen surface now. All right, so let's say this is our C3b. Okay, so now we have C3b that's actually bound onto the pathogen surface. All right, and what's going to happen next is because we have C3b that's bound now, right, onto the pathogen surface, the same thing will happen when we were talking about the fluid phase C3 convertase, is this pathogen bound C3b, right? What's going to happen next is we're going to have factor B come into play again. Okay, so let's draw that now. So remember, we're going to have factor B come into play again. Okay, when factor B comes, it will bind onto C3b. Okay, so what will that look like? We're going to have our C3b that's going to be bound to factor B, right? And do we remember what happens next? Factor D comes, right? Remember factor D, right? So what color do we represent factor D with? Let's go and see here. Oh, so we did factor D in purple, okay, but we'll do factor D in green. All right, so factor D will come in next. All right, so this is going to be our factor D. And remember what factor D did is it went to factor B and it cleaves it into two different proteins, right? Into factor B, into BA specifically and BB. Remember, we don't care about BA because our focus should be on BB, right? So what do we have now? We have, so on the cell surface now, right? On the pathogen surface, we have C3b, which we got because of the fluid phase C3 convertase, right? And then we have BB, right? Factor BB. So this is our BB. So we actually have, what do we have in total? On the pathogen cell surface, we have C3b, BB. This is now C3 convertase, right? But it's C3 convertase that's bound onto the microbial surface, which is important because now what's going to happen is, let's write this, right? This is C3 convertase. And the reason why this is really important is now this whole pathway of converting C3 to C3b and A can get amplified, all right? So what's going to happen is if we have C3 convertase on the pathogen cell surface, right, more C3 can flow by and convert to C3b and A, right? And this can get amplified. So remember, the more C3 that will come, the more C3b and A will be produced, right? The more C3b is produced, the more C3 convertases we can produce, which will again amplify which will then amplify the amount of C3b produced, which will then lead to more C3 convertase on the pathogen cell surface, which will more again amplify, and the cycle will keep on going on, right? So you can form more and more C3 convertases and more C3a and C3b. Now remember what we said in the beginning of the video, C3b has the capability of directly binding to pathogen cell surfaces, right? So if we have another pathogen here, and due to this cycle that's being amplified, right? We have a lot of C3 convertase and a lot of C3b production. Remember, C3b can directly come in and bind onto the pathogen cell surface, which can then help neutrophils and macrophages destroy this pathogen, right? So that's the importance of C3b. And we said C3a, right? Remember, it induces inflammation, right? Induces inflammation. And we also said it helps activate things like histamines, right? So that's basically how C3 convertase gets formed. So let's go down and draw that. So we have let's go and draw that, right? So we have our pathogen cell surface. Okay, so let's draw that in this. Our pathogen cell surface, and we said we have our C3 convertase, which was C3b BB. Okay, so remember we had C3b, okay, and then remember we had factor B, then factor D came in and cleaved B into BB, right? So we have C3b and BB, right? Factor B right here, but it's BB, okay? And remember, we said that C3b BB is C3 convertase. Now, what does C3 convertase do, right? C3 convertase takes C3 and converts it into C3a and C3b. And we said that C3b can then bind directly on the pathogen cell surface, and which can help neutrophils come in and destroy it, right? And we said that the more C3b you have, the more C3 convertases you will form, which will again lead to more C3b production. So the cycle gets amplified. Okay. So, but what can also happen is this: you're forming a lot of C3b, right? What that can do is actually it can go back and bind onto BB, right? Or bind onto what I should say is it can bind onto the C3b BB complex or C3 convertase. So what will that lead to? So if we have our pathogen cell surface here and we have our C3b like this, right? Then we have BB like this, and we're forming a lot of C3b, right? For forming a lot of C3b, that C3b can go back and bind onto the C3 convertase or C3bBB. So if that happens, then what do you form, right? We form C3b, BB, C3b, okay? So it's kind of weird, but that's what we form. So we form C3b, BB, C3b, right? Because remember, this is C3b, right? Then this was BB, and then we're forming a lot of C3b, which goes back and binds onto C3bBB, right? So then we get C3b, BB, C3b. What is this called? This complex is called C5 convertase. And we already know what C5 convertase leads to, right? C5 convertase converts C5 into C5a and C5b. We know C5a induces inflammation and attracts and activates neutrophils. C5b goes on and binds onto what? C6, C7, C8, C9, right? And we know that C5b combined with C6, C7, C8, and C9 and forms a membrane attack complex, right, on the cell surface of the pathogen, which then leads to water influx, leading to osmotic lysis of the pathogen, right, which will lead to osmotic lysis and death of the pathogen. So that's the alternative pathway, right? But what we should talk about is this: if this alternative pathway is occurring, right, what makes it so that this doesn't occur on our own cells? Because this can, when we talked about the fluid phase C3 convertase, right, we said, okay, that's going to help produce C3b. Then we said that C3b will go and bind onto the cell surface of the pathogen, right? But why can't that C3b that's being produced by the fluid phase C3 convertase, why can't that go to our own host cells, right? Our own host cells and bind, and then trigger white blood cells to come and destroy our host cells, or trigger the membrane attack complex to form on our host cells, right? Why can't that happen? So let's talk about that now. All right. So, but before we talk about that, one thing I forgot to mention is remember when we said that we have our pathogen here and we have our C3b, right? And then we have our BB that got cleaved, remember, initially by factor D, which converted factor B into BB and BA. But we have our C3BBB, right? Something stabilizes the C3 convertase here, okay? Something stabilizes this, and that's called factor P, okay? So properidin, all right? So we have factor P here. So properidin is also called factor P. That stabilizes the C3 convertase, all right? Stabilizes our C3 convertase, which is what? C3b BB. All right, so that's important because you want a stable C3 convertase so you can form a bunch more C3b, more C3 convertases, and C5 convertase. All right, if C3b goes and binds again, forming C3b BB, C3b. All right, but let's talk about why this doesn't happen on our own cells, right? So on our own cells, on our host cells, right, we have this amazing regulatory protein. Okay, so let's draw our host cell like this. All right, so this will be our host cell with its nucleus, of course, host cell. And on our host cell, we have something called factor H. Okay, we have factor H. Now, factor H, it turns out, right, is the main regulatory protein of the alternative complement pathway. What does it do? It actually prevents complement activation in our own cells, okay, which is highly important because you don't want your own cells to be destroyed, right, by your own complement proteins. That would be horrible. So it prevents complement activation on our own cells, okay? But then how does it do this, right? There's a few ways of how it does this, and we can quickly go over them. So the first way we can talk about is actually stops the formation of C3 convertase, that being C3b BB, right? So how it does this is this. So let's say, right, let's say you had a person who had dysfunctional factor H. So then when you form C3b via the fluid phase C3 convertase, right? Let's say those C3b molecules started binding on on the host cell, right? When the C3b binds on the host cell, remember what happens next? Factor B comes in, then factor D comes in and cleaves B into BB, right? So then what do we get? We get C3b BB. So this is going to be our BB. Now, with someone that has a dysfunctional factor H, this will occur and you would get the formation of C3 convertase, which would activate complement, increase inflammation, increase the production of C3b, which can then bind on your host cells, triggering it for destruction, right? And it can lead to, of course, C5 convertase formation and the formation of MAC, the membrane attack complex, which can also destroy your host cells. But what factor H does is, if you have functional factor H, it stops factor B from ever binding onto C3b. Okay, so if we remember, first what happens is after C3 binds, we get factor B that comes in, then we get factor D that cleave factor B into BB and BA. But what factor H will do is it's going to compete. Okay? It will compete with factor B, and it won't allow factor B to bind onto C3b. Therefore, you don't form C3 convertase and you don't form C3b, right? That's one way. Another way is, let's say you have C3b has bound it's bound to your host cell. And let's say you did form, or you did factor B was able to bind, factor D did cleave factor B into BB, and now you have C3 convertase, right? C3b BB. What factor H can do is it can come over and it can help dissociate BB from C3b. So it can help dissociate BB from C3b so that you don't form or you don't have functional C3 convertase and you stop the cascade, right? You stop the cascade and the formation of C3b and the complement system overall. So that's pretty cool too. Another thing factor H can do is it can recruit factor I. okay, it can recruit factor I. What's factor I, right? Well, here's what factor I does it, right? What it does is factor I will come over to C3b, okay, to C3b, and it's going to cleave it and convert it into IC3b, okay? So let's say we have C3b that's bound on the cell surface of our host cells. Factor I will come over and cleave C3b into IC3b. That way you can never form C3 convertase, okay? So that's actually pretty cool. So this is also called inactive C3b. So what are the three functions of factor H, or the main three functions that we just talked about, right? One of the functions that we said was it can stop factor B from ever binding onto C3b. Another function we said is it can help kick out factor BB that's bound to C3b, so you have inactive C3 convertase. And another function we said was it can activate and recruit factor I, which will cleave C3b into IC3b, or inactive C3b, which will, of course, that you won't ever be able to form C3 convertase that way, and you stop the whole cascade and the complement system as a whole. Okay, so that was talking about factor H and why the alternative pathway, right? How come that doesn't happen on our own host cells, and we have this nice mechanism that we just talked about via factor H, okay, the main regulatory protein of the alternative complement pathway. What we should talk about next is, right, is how do pathogens take advantage of our complement system? And we can talk about certain examples, all right? So if we talk about Neisseria meningitidis for right now. So let's talk about Neisseria meningitidis, right? Meningitidis. If we talk about this bacteria, we know it can cause meningitis and the whole particular rash and everything, right? The way it can evade this whole mechanism of our complement system is that it can actually bind to factor H. Okay. Now, if it binds to factor H, what will that lead to? So remember, we said that. So let's go and actually draw this. Um, so if we had Neisseria meningitidis, right, the bacteria like this, it actually has factor H binding protein, okay, factor H binding protein that actually will bind. So if we have our like body cells right here, right, let's say this is our own host cell, and we have factor H, right, it will actually bind our factor H. And what that's going to do is it's going to activate our factor H, and the factor H will just do its normal job, and it's actually going to inhibit. It's going to do its normal job, like we said, right? What does it do? It's going to stop factor B from binding to C3b, from binding to C3b. It's going to help kick out or dissociate factor BB from C3b, kick out BB from C3b, and it's going to activate factor I, which will help cleave C3b into IC3b, right? So it's basically going to do everything it can to stop C3 convertase from being formed. Now, that's bad because then if you can't form your complement proteins, right, you're not going to be able to get rid of certain bacteria like Neisseria meningitidis, for example. And then Neisseria meningitidis can go on and infect your cells, right? And so that's bad. So that's one way Neisseria meningitidis can do that, right? Can evade our immune system is it can use its factor H binding protein to bind factor H on our host cells, leading to decreased C3 convertase formation. And if you have low C3 convertase formation, you have low C3 and low C3b, which means um, you can't get rid of pathogens properly. Okay, so and same thing goes with like Borrelia burgdorferi, for example, the pathogen or the bacteria that causes Lyme's disease. It can also bind factor H, right, and which will lead to the decreased production of C3 convertase. And Streptococcus pyogenes, group A strep, remember it has the M protein, right, on it. So the M protein, it turns out, can also bind, right? So the M protein on group A strep can also bind to the factor H on our host cells. And if it does that, again, that's going to lead to decreased activation and formation of C3 convertase, which means decreased formation of C3b, which means, again, you won't be able to get rid of pathogens that well. Okay, so that's how these pathogens can take advantage of factor H, for example. Now, certain pathogens have capsules on them. Okay, so have thick polysaccharide capsules. Now, what are those pathogens? So, if you recall from micro, right, we have certain pathogens like what? Like strep pneumo and Haemophilus influenza, right, that have certain thick polysaccharide capsules. Okay, they contain thick capsules around them. And this actually prevents C3b from binding onto them, from binding onto the pathogens, that being Haemophilus influenza and strep pneumo. And of course, the C3b can't bind to it, then you can't have neutrophils and macrophages phagocytose these pathogens, therefore increasing the count of these pathogens and bacteria, and which will increase, which will help, which will basically lead to more infection from these bacteria, right? Because you won't be able to get rid of strep pneumo and H flu if you don't have, or because due to their capsules, right? Because then you can't have C3 binding to it, C3b binding to it, and you can't, that'll be preventing phagocytosis from neutrophils and macrophages. Because remember, C3b is a potent opsonin. Okay, it attracts neutrophils and macrophages. And you can't do that in this case because things like strep pneumo and Haemophilus influenza have capsules around them, which prevent opsonization. So you can't have neutrophils and macrophages come and destroy it because C3b can't bind to it. Okay. Remember, we were talking about Neisseria meningitidis binding factor H. Well, it turns out Neisseria meningitidis also have, this bacteria also has a capsule that can prevent C3b from binding to it. Okay. So Neisseria meningitidis has both ways of evading the immune system. It has the capsule and it has the factor H binding protein that we talked about. All right. And some other bacteria examples, right, that can take advantage. For example, if we talked about Pseudomonas, right? So write that down. If we talked about Pseudomonas, right, is a bacteria and it can produce things like, it can produce certain enzymes. Okay. And one of those enzymes is C5a peptidase. Right, Pseudomonas can produce C5a peptidase, and it does what it says, right? It can actually cleave and cleave and destroy, or cleave and activate, right? C5a, right? It'll cleave, excuse me, not cleave and activate, it'll cleave and destroy C5a, right? And what will that do, right? If you, since it's a peptidase, right, we're going to cleave and destroy C5a. We're not going to activate it. This enzyme will inhibit C5a, right? Why is that bad? Because remember, C5a did what? It attracted neutrophils to the site of infection. Neutrophils are important for, of course, phagocytosing the bacteria or the pathogen and destroying it. If you can't do that because now you inactivated C5a due to the C5a peptidase produced by Pseudomonas in this example, neutrophils won't be able to be activated by C5a, and less neutrophils to the site of infection means more inflammation and more infection by certain bacteria like Pseudomonas in this case. Okay. So remember we said group A strep, right? Did what? When we talked about that here, we said group A strep, right? Was responsible for, also group A strep also had the ability to evade the immune system, right? By binding onto factor H, right? Remember we said that strep pyogenes, right? Or group A strep, that M protein we were talking about, remember that can bind to factor H, leading to decreased C3 decrease C3 convertase formation. But another thing is that C5 strep pyogenes or group A strep also has C5 peptidase. Okay, so let's go and write that down. Turns out group A strep or strep pyogenes also has C5 peptidase. What does C5 peptidase do? What does C5 peptidase do? It does what? It destroys C5a. This leads to less neutrophil activation and less neutrophils activation, right? Which means less neutrophils will come to the site of infection, which means, of course, this group A strep will continue to infect our cells and increase inflammation. Okay, so group A strep has the M protein that can bind to factor H and activate factor H. And then group A strep has C5 peptidase that can also destroy C5a and lead to less neutrophils to the site of infection. All right. And we can keep on talking about more examples. For example, like Neisseria gonorrhea, right? Or Salmonella. They're resistant to membrane attack complexes. All right. So they can modify their lipopolysaccharide layer or the lipopolysaccharide that's on the outer membrane, remember, right? Or they can modify other proteins, and that can prevent the insertion of C9, for example. Remember, that's the last complement protein to form the membrane attack complex. So if you can't, so remember, you have C5b, C6, C7, C8, and then in bacteria like Neisseria gonorrhea, right, or Salmonella, if you can't if you can't add on C9 to this cascade of proteins, then we can't form the MAC. Okay, so you're not going to be able to form the MAC or membrane attack complex, which means you won't be able to plug this into Neisseria gonorrhea in this case or Salmonella, which means you won't kill or lyse Neisseria gonorrhea or Salmonella. All right, so certain bacteria again, like Neisseria gonorrhea and Salmonella, what's going to happen is they can modify their lipopolysaccharide layer, right? They can modify proteins on their cell surface, or they can modify their lipopolysaccharide molecules, right? Which can lead to the inability to form the membrane attack complex. How? It's actually going to, C9 will have a hard time binding onto these cascade of proteins, right? So then you won't be able to form the MAC and you won't be able to destroy these bacteria like the Neisseria gonorrhea and Salmonella due to this resistance towards the membrane attack complex. All right. So that was basically what I wanted to talk about in terms of pathogen evasion. Now, there is one more thing I want to talk about, and that was, and this is just going to be a quick um, discussion on this, is going to be relating to the kidneys, actually. Okay. So if we talk about that real quickly, because we'll talk about this more on our renal playlist, but there's a certain disease we have to talk about. It's called C3 glomerulopathy. Okay. C3 glomerulopathy can occur due to many reasons, one being what we just talked about, and it's relating to factor H. And if you're deficient in factor H, this can be a reason to have C3 glomerulopathy. Okay, this can lead to that. So if you're deficient in C3, or if you're deficient in factor H, excuse me, sorry. So if you're deficient in factor H, this can lead to C3 glomerulopathy. Okay, how does this work? So remember, if you're deficient in factor H, right, then you can't stop the formation of C3 convertase. So that would mean then you would have a lot of C3 convertase, and you would then have a lot of C3, right? Because remember, C3 convertase, of course, activates and cleaves C3 into C3a and B. So if you have a lot of activation and cleavage of C3, what does that lead to, right? increased amount of C3, right, can start depositing into your glomeruli of your kidney. Okay. And if that happens, of course, we know that that can lead to immune complex formation too. Okay. But this is an important distinction to make because immune complexes, we see that with, so immune complexes, we see that with something called lupus nephritis, right? Which we'll talk about again on our renal playlist, right? It's a complication of systemic lupus erythematosus, right? SLE. But what happens in this is you get a lot of C3 convertase activity, which means you cleave a lot of C3, like we said, into and then into C3b and C3a. So what happens is you actually consume a lot of C3. So if you did labs and you saw the C3 levels, you that would be low, right? So increased consumption of C3. So you would have low C3 levels, but you would have all this excessive formation of C3b and A. And what that can lead to is that excessive formation of C3b can actually deposit into your glomeruli of your kidney. Okay. And obviously this would just be purely C3b with little or no antibodies, right? So if you had a bunch of C3b that deposited in your glomeruli, right? So remember we have our renal arteriole, right? That comes into and then forms our glomeruli, and then you have the renal vein that leaves, right? And then we have our glomerulus here, which is our tuft of capillaries, and then you have your Bowman's capsule like that, and that goes on and forms things like the proximal common tubule, descending loop of Henle, ascending, and so on, right? So let's go and label this, right? So you have your renal artery, or not renal arteriole, you have your renal artery here, your renal vein here, right? And we know that what does that lead to, right? So it's actually going to be your renal artery was going to come in, right? And we know that's going to form your glomerulus, your tuft of capillaries. Eventually, what drains? We know that veins drain, right? Your veins will drain the kidneys and bring it up to your heart, which will go into your circulation, right? So if we actually drew this a little better, so instead of saying that this is going to be the renal vein, we're focusing on the glomerulus, right? So remember, in the glomerulus, we know that the renal artery is what's bringing blood to the kidneys. Then eventually, what happens is this renal artery, remember, it branches into smaller arterioles. So if we go and draw that, right? Well, what we should focus on is this is going to be the afferent arteriole, and then eventually we know that the renal vein will drain the kidneys, right? But what's leaving the glomerulus, right, is the efferent arteriole. Okay, and so what happens is if we get in this factor H deficiency, we have a lot of C3b, right? All that C3b can actually deposit into your glomerulus. Okay, and this can actually damage the podocytes too, because remember, let's say if we have all the C3b, right, that's depositing here, right? So this is going to be our C3b deposition. We know complement deposition increases inflammation, right? And that can later lead to a destruction, right? So it can damage the podocytes, right? Which remember line your the surface right here, right? So remember, this is our Bowman's capsule, and it can destroy the podocytes, which can, of course, lead to proteinuria, which is what we see in nephrotic syndrome too, right? And of course, if this happens over time, we can get scarring, and then that can lead to, of course, scarring, like we just said, but that's also called glomerulosclerosis, right? So that can also lead to glomerulosclerosis, okay? So obviously, factor H deficiency isn't good because it can also affect your kidneys. All right. And of course, if you damage your glomerulus, not only can it lead to proteinuria, but if you damage your glomerulus enough, it may even lead to hematuria also, right? And of course, we know that our kidneys are good for doing what? Regulating blood pressure, right? Through the renin angiotensin aldosterone system. So if you damage your kidneys like this, right, this can trigger the renin angiotensin aldosterone system by increasing renin production, which will eventually increase angiotensin 2 production, as we know, right? And that can lead to the increased release of aldosterone, and angiotensin 2 itself can increase blood pressure too. And aldosterone, we know, increases reabsorption of sodium and water, which can increase blood volume, which can also increase blood pressure. So with this kidney damage, you can get hypertension too, right? Along with proteinuria and hematuria. All right. But we'll talk about all this when we talk about nephrotic syndromes and nephritic syndromes and all that too. All right. So we said one last thing before we end this huge long video. Sorry about that. Is that we said one reason for the C3b deposition was factor H deficiency, right? Because if you don't have factor H, you don't stop the formation and activation of C3 convertase, which means you consume a lot of C3 and convert it to C3b and A, right? Another way you can have persistent activation of C3 convertase is something called C3 nephritic factor. This is an antibody, okay? This is an antibody that actually stabilizes, okay? So it's antibody that stabilizes C3 convertase. C3BBB, right? Remember that? If you stabilize C3 convertase, again, you're going to consume a lot of C3 and convert it to C3b and C3a. And then all this excess information of C3b can go to your glomerulus, and like we saw, can damage your kidneys, right? So that's another way that you can get increased C3b deposition into your glomerulus, okay? And we'll actually talk about C3 nephritic factor when we talk about something called membranoproliferative glomerulonephritis, okay? Which we'll talk about in our renal playlist too. All right, so that's actually it for this video. I'm sorry that was really long. I hope it wasn't all too confusing or anything. I hope this helped even a little bit. Thank you guys for watching and see you guys in my next video.